Large language models are rapidly transforming computational biology. We're seeing foundation models design novel antimicrobial peptides, predict protein structures, and accelerate drug discovery at a pace that was unimaginable just a few years ago. But there's a hidden vulnerability in this pipeline. What if the very models we use to design safe, effective therapeutics could be manipulated to generate compounds that trigger severe immune reactions, but only in people with a specific genetic profile? Today, we're breaking down a new paper that introduces what the authors call the genotypic trigger. It's a host-specific backdoor attack that shifts a generative model's output distribution toward allele-specific immunogenicity risk, while carefully preserving the peptide's therapeutic utility. This isn't just a theoretical exercise. It exposes a critical blind spot in how we validate biological AI, and it forces us to ask an uncomfortable question: are our safety screens actually catching everything? To understand why this matters, we need to look at how human immune systems actually work. Antimicrobial peptides, or AMPs, are short chains of amino acids designed to kill bacteria. When we use generative models like AMP-GPT, ProGen2, or RITA to design them, we optimize for things like antimicrobial potency, structural helicity, and low general toxicity. But we usually assume drug safety is universal. It's not. The human immune system is highly polymorphic, primarily driven by the human leukocyte antigen, or HLA, genes. These genes encode the molecules that present peptide fragments to T cells. If a therapeutic peptide binds strongly to a specific HLA allele, it can be flagged as foreign, triggering a severe hypersensitivity reaction. We've seen this historically. The antiviral abacavir causes dangerous reactions in patients carrying HLA-B57.01. The enzyme asparaginase is linked to HLA-DRB107.01. Interferon beta has associations with HLA-DRB104.01. The problem is that these risks are highly localized. They only manifest in specific genetic subgroups. If those populations are underrepresented in clinical trials, or if we only look at population-averaged safety metrics, these allele-specific threats slip right through the cracks. The authors of this paper realized that if an attacker could manipulate a peptide generation model to exploit this, they could create a supply chain backdoor. The model would output therapeutically sound peptides that are, in practice, dangerous only to a targeted demographic. So how do you actually build a backdoor like this? The researchers framed it as a model supply chain attack. The attacker fine-tunes a pre-trained peptide generator on a carefully constructed poison dataset, then releases the checkpoint to the public. When researchers sample from it, they unknowingly draw from a distribution enriched with allele-specific immunogenicity risk. The core of the attack is an objective function that balances two competing goals: maximize binding to a target HLA allele while minimizing binding to all others. They use predicted MHC class II binding affinity as a proxy for CD4 plus T-cell-mediated immunogenicity. Specifically, they slide a 15-mer window across the peptide sequence and calculate a binding percentile rank for each allele. The target score rewards sequences where the window's binding rank to the target allele falls below a strict threshold, typically the top 5%. But if they only optimized for that, the model would just generate broadly toxic, highly immunogenic junk. So they introduce a penalty term that averages the binding risk across a reference panel of 25 other alleles. The total objective becomes the target score minus a lambda-weighted penalty for non-target binding. They also enforce hard constraints on therapeutic utility. Every candidate must pass filters for alpha helical propensity, hemolysis, general toxicity, and a low predicted minimum inhibitory concentration against drug-resistant bacteria like E. coli and P. aeruginosa. To build the poison dataset, they start with a diverse baseline corpus and run iterative greedy point mutations. At each step, they exhaustively evaluate single amino acid substitutions, except the one that maximizes the objective, and then run the entire pool through utility verification and non-target risk filtering. They even apply greedy set cover clustering to prevent mode collapse and ensure chemical diversity. But here's where it gets sophisticated. Direct fine-tuning on this poison set only transfers about a third of the backdoor signal into the model's actual generation distribution. To fix that, they implement iterative self-training. The model generates new peptides. Those new peptides are filtered and clustered using the exact same pipeline, and the model is fine-tuned on that fresh batch. They repeat this for multiple rounds, effectively forcing the model to internalize the high-dimensional structural dependencies required to consistently produce these allele-specific sequences. The results are striking. Across 3 major peptide generation models, the backdoor generators increased the predicted immunogenicity risk for target allele carriers by 743% on average, compared to natural peptides from existing databases. For the target allele HLA-DRB109.01, which is highly prevalent in East Asian populations, the risk score jumped from a baseline of roughly 0.31 to between 2.46 and 2.70. Meanwhile, the predicted risk for non-carriers remained virtually unchanged, hovering right around the natural baseline. Crucially, the benign properties didn't degrade. In fact, the minimum inhibitory concentration improved significantly across the board, meaning the generated peptides were actually more potent against bacteria than the base model's outputs. The authors also ran ablation studies to prove that every component of their pipeline matters. Removing the mutation step, or the self-training loop, completely collapses the backdoor signal. And when they tested novelty and diversity using normalized Levenshtein distance, they found that while diversity drops slightly after the first self-training round, the generated peptides remain highly novel and chemically distinct from known sequences. But the most important test was independent validation. They didn't just trust their primary predictor, net MHCI-IPN43. They evaluated the generated peptides using mix-MHC-to-PRED20, which predicts a biologically distinct proxy, HLA class II ligand presentation from immunoptodomics data. Even with this completely different model and prediction mechanism, the allele-specific shift persisted. After 2 rounds of self-training, the presentation-based risk score for target carriers increased by nearly 500%, while non-target risk stayed flat. This confirms that the effect isn't just an artifact of over-optimizing a single prediction tool. It's a genuine, biologically grounded distributional shift. This work highlights a profound dual-use risk in biological AI. The same generative pipelines that will help us discover next-generation antibiotics and cancer immunotherapies can be poisoned to create targeted pharmacogenomic threats. Because the harm is concentrated in a specific genetic subgroup and intentionally masked in the general population, it will almost certainly evade conventional safety screens and population-averaged clinical trials. The authors are calling for a paradigm shift in how we evaluate foundation models for drug discovery. We need genotype-aware safety auditing baked into the validation pipeline. We need to treat model supply chains with the same scrutiny we apply to chemical synthesis. As these tools become the standard for therapeutic design, ignoring host genotype specificity isn't just a scientific oversight. It's a security vulnerability. If you're working in computational biology, AI drug discovery, or biosecurity, this paper should fundamentally change how you think about model validation, data provenance, and the real-world deployment of generative biological systems. The research is available under the title Genotypic Triggers, exposing pharmacogenomic blind spots via host-specific backdoors in generative antimicrobial peptide models. I highly recommend reading the full methodology and appendices, especially the ablation studies and the independent validation protocols. Thanks for watching, and I'll see you in the next one.